Hi, my name is Nurishada binti Shahrul. Today, me and my group will be presenting on a topic which plays a critical role in forensic science, which is about burn bones as source of DNA for human identification. When people die in events like fires or explosions, their bodies are often burned so badly that normal identification methods don't work. In these cases, scientists try to identify the remains using DNA from bones, but getting DNA from burn bones is very difficult because heat damages the DNA. Although new DNA testing methods have improved over the years, burn bones are still a big challenge, and this article looks at what is currently known about how burning affects bones and how we can get the most DNA from them. The objectives of this research are to understand what happens to DNA in bones when they are burned. The second is to find out which bones and which techniques give the best chance to get usable DNA. And the third is to highlight gaps in current research and suggest areas for future improvement in forensic DNA recovery from bone remains. The main problem stated in the research is that burn bones undergo physical and chemical changes that make DNA very difficult to extract. High temperatures can damage the DNA or cause it to bind tightly with the bone minerals, reducing chance to successfully recovery. While some techniques used for ancient bones show promise, they may not work the same way on modern burn remains. There is also limited understanding of how heat affects DNA in bones, and no standard method exists for extracting DNA from burn bones in forensic science.
Assalamualaikum and hi, my name is Siti Saratinisa binti Abdul Talib. My metric number is 2022674988. In this video, I'm going to talk about sample collection. So when we try to get DNA from burn bones, picking the right one is super important because some bones hold DNA better because they are denser, meaning they are tougher and protect the DNA inside. For example, teeth are great because they have hard enamel that shields DNA, and big leg bones like the tibia or femur are strong and weight-bearing, so they often survive fire better. There is also a special part of the skull called the petrous bone, part of the temporal bone, which can have 4 to 16 times more DNA than teeth in some cases. On the other hand, small bones like those in hands and feet often burns up completely because they are tiny and not as protected by muscle or fat. When a body is burned, muscles shrink fast from the heat, pulling the body into a boxer pose, like a fighter with clenched fists. This can break smaller bones, making them bad for DNA. Plus, other things like weather, animals chewing on bones, or being buried can damage DNA too. Scientists say there are 3 ways bones break down, which is chemicals hurts the soft parts, the hard mineral parts wear away, and tiny microbes sneak in and mess things up. Next, for bone analysis, before we try to get DNA, we can check bones to guess if it will work. By looking closely, we can see clues. For example, the color and feel of a bone can tell us how hot and long the fire was. Some studies, like ones by Walker and Fredericks, show that bone color might hint at whether DNA or collagen, a protein in bones, is still there. Lighter or darker shades could mean more or less damage. Under a microscope, we look for tiny cells with nuclear or bone patterns called osteons that suggest DNA might be present. When bones get to 300 or 400 degrees Celsius, all the soft organic stuff vanish and hard crystals start to form in the bone. Above 500 degrees, these crystals, called hydroxyapatite, grow really big, which can trap DNA and make it tough to get out. So, just by looking at bones with our eyes or toes, we can get a sense of whether DNA might still be there to help with the identification. Next, I will talk about sample preparation in tissue sampling. Our goal is clear: collect bone tissue to extract DNA for identifying incinerated remains. When a fire happens, it can destroy a lot, but bones might still hold DNA clues. By carefully gathering samples, we aim to get enough good DNA to figure out who someone was, helping police and families solve cases after tragic events like fires. Here is the big challenge. Heat will degrade DNA, reducing both its quality and quantity. fire gets super hot, breaking down the soft parts of bones and the DNA inside. This makes it tough to find enough usable DNA, and what's left might be damaged or in tiny amounts. We have to be smart about how we collect it to beat this problem. And why is this a big deal? Effective sampling increases the success of genetic analysis for forensic. If we pick the right bones and handle them well, we are more likely to get DNA that works for testing. This can help identify people, bring closure to loved ones, and support justice in tough cases. Good sampling is the first step to making it happen. Next, I will quickly guide you through tissue sampling from burned bones for DNA and identification. Start by picking strong bones like hip, tibia, femur, or the petrous bone from the skull because they are dense and hold more DNA after a fire. But skip small, weak ones like in hands or feet since they burn up easily. Then carefully cut a small 1 to 2 gram piece with a clean saw or tweezers, avoiding damage, and place it in a label tube. Watch out for issues like heat, weather, animals, or burial that can ruin DNA. So keep sample clean because our goal is to get enough DNA to test and identify remains for forensic cases.
Assalamualaikum. My name is Nur Ainaja binti Edi Muhammad. My metric number is 2022458464. So in this video, I will explain the sample preparation for burn bone. So this is the second step or the other step that we can do for extract DNA from the bone. So the second step is demineralization, which is demineralization is the process of removing the substance from the bone, which is mineral. So why this is important is because we want to avoid mineral degraded in the bone. So we try to extract first. We need to remove the mineral first to extract the DNA for further analysis. Okay, so for the demineralization, EDTA was used as a demineralization agent. And then the combination of demineralization and cryogenic pulverization is good because it helps to enhance more DNA and it also helps to avoid the contamination. And last, this process should be happen more longer because the more time can help to enhance more DNA.
Alright, next is DNA extraction. So in this study, they use silica-based technique to extract the DNA from the sample. In this technique, there are 3 steps, which is bind, wash, and elute. So the DNA will denature and then bind to silica particle, and then it will separate it from inhibitor and non-target material. To get the target material, we need to wash the sample with the alcohol. This method was used because of it more cost efficient, easier, quicker, and it's help better for removal DNA inhibitor than other method that exist. Okay, the process of silica-based extraction have 2 general approach, which is suspension in a silica solution and kit-based binding to silica beads or membrane. So silica suspension is a common alternative to silica binding that allow for processing of substantially larger sample volume than commercial kits. So if we use this technique, we can extract more DNA because of larger sample volume that we use.
Assalamualaikum. My name is Umi Safainada binti Abu Bakar. So I will be present the recommendation for the study of bird-born as a source of DNA for human identification. So for the first one is develop standardized protocols for DNA extraction from modern bird samples. Well-established protocols exist for fresh and extinct bone samples, but modern bone samples lack standardized method. Decisions are often made case by case, which can lead to inconsistent results. Developing a universal protocol would improve reproducibility, reliability, and efficiency in forensic investigations involving burn remains, such as in fire-related mass casualties. The second one, investigate DNA yield from non-standard skeletal sampling sites after extreme tissue damage. Certain bonds, like the vitreous part of the temporal bone, are known to protect DNA better in severely burned remains where common sampling sites, like teeth, may be too damaged. These alternative sites might still preserve visible DNA. Investigating this could explain the available options for DNA recovery in degraded samples. The third one, conduct comparative studies on DNA yield from different skeletal sites in incinerated remains. Although studies like Colston have evaluated DNA content across skeletal sites, they didn't focus on incinerated bonds. Performing similar comparative analysis under bond conditions would help establish which bonds are most reliable for DNA recovery after fire exposure. Next, establish a graded system for DNA yield from incinerated bonds. A rank or graded system indicating the likelihood of DNA presence in various burn bond types would help forensic teams prioritize which bonds to sample first. This would streamline the identification process, saving time and resources in mass disaster scenarios. And refine downstream DNA processing techniques for burn samples. Even if DNA is successfully extracted, it may be degraded or present in low quantities. Improving amplification and analysis methods tailored to the quality of DNA in burn bonds could enhance identification accuracy and success rates. And the last one, we could apply conservative DNA extraction techniques to incinerated samples. Many methods used for degraded archaeological remains might also work for burn bonds but haven't been tested in that context. Exploring their effectiveness on incinerated remains could reveal new, more effective strategies for DNA recovery. So that's all from me for the recommendation.
In conclusion, burn bones can still hold valuable DNA for identifying human remains, but extracting the DNA is much more harder compared to the undamaged tissue. Currently, there is no standard method specifically designed for burn modern bones, while techniques used for ancient bones such as EDTA demineralization and silica-based DNA extraction show promise. They may need to be adjusted for forensic cases involving recent burns. More research is needed to understand how heat affects bone structure and DNA survival to determine which bones provide the best DNA after burning, and also to create faster, more reliable methods for use in real forensic investigations. A combined approach using knowledge from biology, chemistry, and forensic science is essential to improve DNA recovery from burnt human remains. That's all from us. Thank you.